Inside you, there's this world of tiny machines called proteins. There's over 30,000 different kinds of proteins, and each one has a complex shape. Some proteins use their shape to break apart molecules, like turning food into you. And other proteins send messages by fitting into an exact shape, kind of like a lock into a key. Proteins can even change their shape and pass on messages to other proteins. When something goes wrong in your body, it's often a case of a protein struggling to do its job. But we don't know which protein. It's like a jungle in there. Each of your cells has millions of proteins, and they're all bouncing around, dancing together. We need this way to find a needle in the haystack, to find which one shape of all these shapes is struggling, so we can pinpoint where disease happens. My Ph.D. work is a search for proteins, and I find them using this really cool technique called DGAY. DGAY is a way to separate proteins and then compare two groups of proteins, so you can focus just on the differences between them. So here's how it works. Each protein has a mix within it of positive and negative charges. And the thing about proteins is those charges change depending on the surrounding environment. In an acidic environment, proteins become more positive. In a basic environment, they become more negative. So we set up an environment that moves from very acidic to very basic, and we run through this electric current. So the electrons are like zipping through, pushing the proteins through the strip. And as the proteins travel from acidic to basic, their charges change. They get a little less negative. When a protein reaches its neutral point, it stops. It doesn't have a charge, so it's not affected by current anymore. So what this does is it physically separates all of our proteins by one of their chemical properties. A second way we can separate proteins is by size. We make them run a race, a sort of molecular obstacle course. And because proteins come in all these different shapes, we have to first unfold them to make it a fair race. The small proteins move through very quickly, they dart through the holes, and the bigger proteins, they don't get as far. They move slower and they get tangled up more easily. So what we've done is separate proteins by two chemical properties, by charge and by size. But we actually don't do this just for one sample. We do this for two samples at the same time. So in the same gel, we're running sample 1, which could be from a disease state, and sample 2, which could be from a healthy state. And the key innovation after that is this pair of dyes that stick to all proteins. And these dyes can survive the whole process, and they don't affect the result of any of the races for any of the proteins. So what that means is we can run two groups of proteins at the same time on the same gel. And in the end, they'll be labeled different colors. One group will be red, and one group will be green. So where the two groups overlap, their colors combine to make yellow. Those proteins are like in sync. They're the same between both groups, and most proteins overlap. But occasionally, one group will have more of a certain protein. It's like that protein is louder. And this technique lets us see that. We'll see a spot that's tinted a little more to the red or a little more to the green. It diverges from yellow. Occasionally, a protein can get modified or decorated, and the chemical change will cause it to run one of the races differently. For example, maybe it'll become lighter, so it'll run the size race a little faster. And we can see that too. The yellow spot will separate into a green spot and a red spot because those proteins have slightly different chemical properties. And this is essentially how we find a needle in the haystack. We can focus just on the differences between a healthy group and a disease group. We can find which protein is struggling to do its job, so we can build new treatments to help it. And that's how DGAY works. And it's the basis of my PhD.